All right, our next presenter is CDR3 Therapeutics. Matthew? Excellent.
Thanks, Matt.
Thank you.
Matthew Lorence with CDR3 Therapeutics, and we're working on stem cell-based immunotherapies. Okay, let me move that. Is that better? No? Is it not on?
Should be.
Yeah.
Are we picking it up? That's
No.
good, yeah.
If I have to do that, I will. Okay.
Take matters in your own hands.
Yeah, I can hear it now. So the T-cell immunotherapy market is expected to reach over 17 billion in the next few years, but since 2017, there's only been 6 FDA-cleared therapies. And all of these are for liquid tumors, B-cell-based cancers. There have been no FDA-cleared therapies directed against solid tumors. Why is that? And that's because the engineered cells just don't persist. Under the current method of expanding the cells outside the body, these engineered T cells are gone within weeks. And to be effective against solid tumors is really going to require prolonged persistence. Another somewhat unrelated fact is that these therapies also target cells or proteins that are normally expressed on the cells, so they can lead to a severe condition known as cytokine release syndrome. So CDR3 is looking at it from a different perspective. Instead of working at sort of the end of the T cell production, which is the current technology, where blood cells are harvested from the patient, the CAR or T cell receptor gene is added, and then those cells are expanded outside the body, then reinfused into the patient, where they can become damaged and exhausted so they are not effective. We actually start at the beginning of the spectrum with hematopoietic stem cells from the patient that are harvested. The TCR or CAR are added there, then those cells engraft in the bone marrow of the patient, where they provide a persistent, self-renewing source of normally developed T cells, which then can be effective, unlike the ones that are currently engineered. Our first target indication is HPV-associated cancers. These cancers now actually represent about 5% of all cancers worldwide. And whether they're cervical, oropharyngeal, or anal, the problem with these cancers is once they become metastatic, they're incurable. So there is a good market opportunity here for this technology and this indication. We're currently seeking to raise a little over $14 million to fund a seed round. We've laid out the milestones and amounts of money that we need to get there. We have at least 2 follow-on investors that are interested, so we're currently looking for a lead investor to get us to our seed round. Our team who founded the company are all professors in the School of Medicine at UCLA, and together they have over 75 years of experience in immunology and have raised more than $100 million in grant funding. So in summary, CDR3 technology introduces CAR or TCR genes into minimally manipulated patient HSCs instead of peripheral blood cells. They engraft into the patient's bone marrow and produce healthy cells retargeted against the cancer. They even produce other bone immune cells, including NK and macrophages that will target against the cancers. We also target foreign proteins instead of cell proteins to reduce the risk of CRS. We have completed some proof of concept studies in non-human primates showing that technology is safe and non-toxic. We've seen formation of these engineered T cells in many different anatomical sites as expected, and the engineered T cells have been shown to be reactive against both HIV and in the laboratory because HIV was the model for the proofs of concept. We currently have in vivo studies in progress with humanized mice to validate efficacy of the HPV TCR that we currently have. And we consider this to be a platform technology because the same conditioning protocol and treatment protocol will be used, and the only thing that we need to vary is either the TCR or the CAR that's added. And again, we're seeking a lead investor to fund our $14 million seed round to support an IND submission and a phase 1 clinical trial for HPV-associated cancers. Thank you.
So to be clear, the therapy would involve taking a bone marrow sample from the patient manipulating that and then regrafting that into that patient's
Well,
bone
we
marrow?
mobilize HSCs from the patient and then modify those, and then the conditioning treatment is very mild because we don't have to eliminate the bone marrow, just make a little bit of a space in the bone marrow for these modified cells to engraft.
The innovation from your company, would you say it's mostly around the process, or is it around the CAR or the targeting element?
It's kind of both. So it turns out to be not very easy to introduce CARs or TCRs stably into stem cells and have it persist. So we've been able to, through trade secret work, basically considering these trade secrets, understand some of the key structural requirements for that CAR or TCR to be able to stably present. And at the same time, we've also created some CARs and some TCRs around this.
Go for it. You guys have tested for this persistence after the stem cell the HSC differentiation?
Well, that's what the non-human primate study showed,
Got it.
that it persisted for at least 2 years before the animals were sacrificed.
One of the issues with persistence is that there's no off switch, right?
Yeah.
Any thoughts on that?
So that is a potential concern. One way of looking at it is maybe from the opposite side. If these T cells are persistent, it's in some ways almost like a vaccine against the indication because they're there all the time to keep the tumor in check. But again, in the non-human primate studies, after more than 2 years, there were really no visible issues or concerns with the fact that those TCRs were persistent. Sorry, those CAR cells were persistent.
Would this be a one-time sort of
Yeah, it'd be a single treatment.
a single treatment, which would last for 2 years, more or less?
Should be lifelong. There's no reason for those engrafted T cells to stop producing those engrafted HSCs to stop producing T cells. The non-human primate studies only were carried out, as I said, a little over 2 years, so we don't really know what the persistence should be, but there's no reason to believe it would end.
So how would you compare your approach compared to allogeneic T cell, like off-the-shelf T cell treatments, like T-scan and Sana, and companies that are trying to move away from autologous technologies?
I think allogeneic therapies are a great idea that have not yet come to fruition. So right now, what really should work, at least what's been demonstrated, is autologous. And specifically with patients that are suffering from cancer or HPV-associated cancers, this probably is a much faster treatment. I mean, those cancers aren't that aggressive, although they are fatal once they're metastatic. I don't know if that answers your question.
I mean, I guess what is the advantage of traditional T cell therapy? Is it because yours like once treatment and that's it? And there's, CABA, not in oncology, in lupus, they have shown that there is promise for, clinically, for allogeneic T cell treatment. And then the other side, and also Adapt Immune, I don't know, like there's a company that is developing T cell treatment for solid tumors. I'm not sure if I know they got approved recently in Europe, and I think they're seeking approval in the FDA. So there is also autologous T cell treatments that are being advanced for solid tumors, and one of them either approved or soon to be approved.
Okay, I wasn't aware of that, sorry. Any other questions?
You mentioned that you're using trade secret protection to kind of protect the specific engineering that you're doing.
Basically, the construction of the TCRs and CARs.
Got it. And so, is there any patent protection?
Yeah, we have two patents that were filed by the co-founders that were licensed exclusively from UCLA, and a third one that's pending.
And are those covering this general platform of, any TCR or CAR into an HSC? Or?
So that's far too broad to get patented,
Yeah.
but it covers different aspects of the process.
Got it.
And how long will, or how much, the 14.1, it will take you to the end of your phase 1 or at the beginning of your phase 1?
The total amount should get us to the IND submission in the beginning of the phase 1.
So a start of your phase 1. And will you qualify for like the new guidance from the FDA, single pivotal, single arm approval for cancer? Is that something that you can take advantage of in your planning?
I can't really answer that. We do have a regulatory consultant, and I don't have the expertise in that area, sorry.
And how much money did you raise to date?
Nothing.
Okay, so this is your first.
Completely self-funded.
Oh, wow.
And either NIH grant or self-funded by the founders. All right, thank you very much.